So good morning, everyone, and welcome to this talk. As you all know, mutations in DNA have consequences in the functionality of proteins that are the final effectors of the instructions contained in our genome. And however, protein function can be altered by post-translational modifications in response to physiological but also pathological situations. This is relevant in pathologies where no DNA mutation could be associated with the development of the disease. However, to study these modifications in different pathologies is harder and definitely much more expensive than studying their genome. Our today's speaker is an expert in a type of protein modifications that is the conjugation with ubiquitin and ubiquitin-like molecules. He has published many articles about this topic in the most prestigious magazines. Dr. Xirodimas received his undergraduate degree in molecular biology in 1996 from the University of Dundee in Scotland, and he continued his formation there, working in two different laboratories. In 2005, he was the recipient of a fellowship from the Association for International Cancer Research to develop an independent research team in the Center of Gene Regulation and Expression at the University of Dundee. In 2011, he moved to France, and there he was recruited to be a research group leader in Cell Biology Research Center in Montpellier in France. So today, Dr. Xirodimas is going to tell us about the relevance of ubiquitin-like modifications in cancer and its possible exploitation as therapeutical targets. Well, Dimitris, thank you for attending our call, and well, your audience.
Okay, thanks a lot, Carlos, and thanks also to Natalia for organizing this meeting. It's a really great pleasure to do this, even if I would prefer to come Santander because I have personal connections there and I love, North Spain. So, but now it's the way that we have to do things. So thanks very much for the invitation. So as Carlos said, I mean, our group is interested on post-translational modification of proteins with a small family of proteins called ubiquitin and ubiquitin-like molecules. And today I'm going to focus, I'm going to give a more broad introduction to the field, and I will try to give you some overview of our research on the ubiquitin-like molecule Netdate and how we can use this pathway for therapeutic intervention with focus in cancer. So this is highlighted highlights the basic sort of dogma in biology, where from DNA go to RNA. You make proteins where they are the workhorse of in cells that they execute all the functions in the cells, and you elicit the biological outcome. But now we know that these pathways are constantly challenged by actually intrinsic or extrinsic stress, which we define now as genotoxic or prototoxic stress. So basically, damage that happens to DNA or RNA, or damage that happens to proteins. So cells have developed a highly sophisticated processes in order to detect and repair, and in the case that the repair cannot happen, to eliminate both DNA or protein damage. And this is very critical because if that doesn't happen, if you sustain both protein damage and DNA or RNA damage, this goes to pathology. And of course, you will know that DNA damage can cause to cancer, where protein damage is well established for the creation of new generation. But also, I will just show you that also protein damage can be involved in cancer pathology. So now we know that this family of ubiquitin and ubiquitin-like molecules are absolutely essential in all these processes of how the cell detects, repairs, and eliminates, damage from the cell. So, as Carlos said, I mean, so this family belongs to over 200 types of post-translational modifications that exist in cells. But the sort of unique aspect of these post-translational modifications is that here we're discussing about a small protein that is convergently modified to another protein. So normally the post-translational modifications that we are all more aware of, they are all sort of chemical or lipids that are modifying proteins. In this case, we have a protein that is modifying another protein. And this also, from an evolutionary point of view, it's quite interesting because these molecules themselves, they are actually subject to evolution and changes. So you can imagine that these molecules can actually change, if you like, the biology with evolution, which is in contrast, for example, with phosphate groups that basically they are, as far as the evolution is concerned, they're evolutionarily dead. They cannot, they cannot change. Now, the key question now, we know that these pathways, for example, if we take, for example, ubiquitin, they're involved in the regulation of every biological process you can think of. And if there's something that is not controlled or we don't know, it's probably because it has not been studied. So these pathways are really involved in the regulation of every biological process. And the key question here is how a single protein can be involved in the regulation of so many biological processes. So if we take ubiquitin as an example, and this is quite complex, but I will try to go through a bit in a simplified manner, is that ubiquitin, as I said, can modify post-translational modify other proteins as a single moiety. But also, because ubiquitin is a protein itself, it can also modify itself. So it can cause what we define now the ubiquitin chains. And what is interesting here is that depending on which sort of residue you use on ubiquitin to make this change, you create a completely different signal for the cell that is involved in a completely different biological outcome. So here I just give you like a very sort of overview of summary, if you like, of biological processes controlled by different types of ubiquitin chains. But I guess the one that you're most aware of is that the function of ubiquitin chains to cause or to target a protein, a modified protein for proteasomal degradation, to eliminate this protein, the modified protein from the cells. So here I will just describe a bit of how these ubiquitin molecules, they're really involved in elimination of proteins from the cell, what we call in actually as the protein quality control system, and how ubiquitin is impinging of controlling the state of the proteins in the cell. So as I said that the cells are constantly exposed to stress, in this case prototoxic stress, and here I just highlight a few examples. Even DNA damage that we know is inducing protein damage. Chemotherapeutics that are used in clinics, they also protein damage, oxidative species or elevated temperature. And the key characteristic is that all these stresses, what they do is that they're causing the misfolding of native proteins. And here the system has to detect and repair this protein damage. So there's a whole sort of family of proteins called the chaperones, which they detect these misfolding proteins, and either they try to refold them back into the native state, or if not this possible, they're using the ubiquitin system in order to modify these misfolding proteins with what I said, the ubiquitin chains, and this now acting as a signal to degrade these misfoldings in the cell. So the proteasome is one of the key machineries that degrades, that recognize ubiquitinated proteins and degrades them, and also the autophagy system. Now we know that it can also be involved in the clearance of misfolded proteins. So of course this is critical, because if these proteins, misfolded proteins, they're not sort of degraded, eliminated, they will accumulate potentially as aggregates in the cell. And now we know that the formation of aggregates, irreversible aggregates, insoluble aggregates of misfolded proteins, they are involved in pathology. They are regarded as the hallmark of neurogenetic diseases. So again, you can see how the ubiquitin system is critical in order to prevent sort of this pathology. But what is also now clear is that also in cancer, the creation of misfolded proteins is also part of the pathology. So, for example, as Carlo mentioned, many mutations that they are found in cancers. One of the things they do is they're causing protein misfolding. And what happens is that in this case, when you increase the percentage of protein misfolding in the cell, you increase the load of proteins that has to be degraded. So basically, there is a great dependency on the proteasome to degrade and increase loads of misfolded proteins. So this is why we believe that many cancer cells, they're extremely sensitive to the proteasome function, because now the proteasome is oversaturated, if you like, from the creation of misfolded proteins due to the mutation, and now these cancer cells, they're becoming they are super sensitive and depend a lot on the activity of the proteasome. And this, if you like, provided, if you like, initially the evidence that by blocking the activity of the proteasome, we can actually create now a specific cancer therapeutic approach. And indeed, the compound that probably bortezomib or Velgate, is a specific inhibitor of the proteasome that blocks the degradation of proteins in the cells. It's a reversible inhibition. It was approved back in 2003 for treatment of patients with relapsed or refractory myeloma. I have to say that the clinical trials had to stop prematurely because the compound was so effective that it was regarded as unethical to keep the clinical trials on. So it was a super fast, it was like basically like the vaccines that we have now for COVID, that they went through the clinical trials extremely fast because of their success and of course of the pressure that we have in society. So now these compounds, this Velgate is now an approved and a commonly used drug for multiple myeloma and mantle cell lymphoma. But something interesting about this compound, and maybe we can discuss this later, is that it doesn't seem to be so effective in solid tumors. So now people are trying to understand why this compound is so effective in this type of tumors, but not in solid tumors. But nevertheless, this provided a very strong proof of principle that by targeting the ubiquitin system, you can actually devise therapeutic approaches, for cancer. Now, ubiquitin it's one of the members of a growing family of proteins, which we call them ubiquitin-like molecules. So these proteins are very similar to ubiquitin. So they have very similar structure, but of course they have their own independent functions. So here I just highlight a few. The ones that they're more sort of studied at the moment is SUMO, Netdate, and IG15, and of course ATG5, which is involved in autophagy. Now, the key thing about these molecules is that they have, if you like, their own unique conjugation pathway in order to modify substrate of industry in this case. So here just highlight that this conjugation, these proteins are modifying substrates using the three key enzymes, which we now refer as E1, E2, and E3. So these are three enzymatic activities that they pass, in this case, say ubiquitin, from one enzyme to the next, till eventually the E3 enzyme, which is we call the E3 ligase, is the one that directly binds to the substrate, the covalently attached ubiquitin on the substrate. And what is critical here is to mention is that each molecule has its own unique enzymes for modification of the substrate. Now, it's based on the success of the bortezomib, Velgate in clinical as a cancer therapy. Now, many pharmaceutical companies they decided to target all ubiquitin-like molecules. So now there's a series of inhibitors that specifically inhibit the E1 enzymes, the first enzyme in the cascade of each of this pathway. So basically, these inhibitors, what they do is they completely block the conjugation of, in this case, of ubiquitin or Netdate or SUMO, etc. So all these compounds now, they are in clinical trials now. But what I will focus today is on the ubiquitin-like molecule Netdate, and particularly on this compound, the inhibitor of this system, which is called MLM494 or pevonedistat, which is its clinical sort of name, and how potentially we can use these compounds in cancer. So first, I will just give you a bit of introduction of the Netdate pathway. I will give you an example of how, like a bit of basic research of how potentially the Netdate system can be involved in cancer development. And the second part of the talk, the last part, will be how potentially we can use a strategy to use these inhibitors in the clinic. Now, so the Netdate pathway, as I said, it has its own E1, E2, and E3 enzymes to conjugate substrates. And now we know that the proteins that are modified by Netdate, they're belonging to two different classes. We have the Calin family of proteins. So these are a small family of proteins that belongs to an E3 ubiquitin ligase family. So they are scaffold proteins. And when they're modified by Netdate, they have increased ubiquitin activity to degrade proteins for proteasomal degradation. So in this case, what Net is doing is stimulating ubiquitination of proteins through modification of these protein, calins. But also now we know from proteomic studies that we've done ourselves and other groups that there's many non-Kalin, as we call them, substrates, which now we think there are more than 1,000. And also, I know that Carlos is quite interested in this to develop sort of really innovative approaches to specifically identify Netdate substrates in cells, because that's critical if we want to understand how this pathway really works and how inhibitors of this pathway elicits, if you like, anti-tumor activity. So, of course, because what we knew at the beginning is that the Netdate pathway somehow, as I discussed, it can control the degradation of proteins, and based on the success of Velgate, the companies decided to develop an inhibitor for the Netdate pathway, which is now in phase 3 clinical trials for multiple myeloma and also solid tumors. And what this compound does is specifically inhibits the E1 enzyme for Netdate. So basically completely blocks the Netdate pathway in cells. Now, what is also critical to understand here is that these pathways, they are very dynamic, so what I described so far is that how these molecules, they are, through the activity of the E1, E2, and E3, they're modifying substrates, but the reaction is irreversible. I mean, similar to phosphorylation, where you have the kinase and the phosphatase, the system has the so-called deconjugating enzymes, which basically they remove the molecule from the substrate in order to release, in this case, NED from the substrate, and you can reinitiate the cycle of conjugation. So for NED, we have two types of enzymes, of deconjugating enzymes. We have the COP9 signalosome and NEDP1, and now we know that the COP9 signalosome is dedicated to remove Netdate from Kalins, where the NEDP1 is mostly specific in removing Netdate from non-Kalin targets, all the 1,000 substrates that I just described to you. Now, an interesting thing about NED as well is that it's similar to ubiquitin and also can make this Netdate change. And now we have evidence, and I will show you some data in a minute, that the pathway is upregulated in a variety of cancers, which is highlighted here. So it suggests that maybe the increase of NEDylation may act as an oncogene, if you like, in these tumors. And of course, its inhibition by an inhibitor might have an anti-tumor activity. So key questions that we want to understand in the lab, and I guess in the field at this moment, is that what is the molecular basis of the NEDylation defects in cancer? So why Netdate is increasing in cancers, and if there's any system that basically can detect this misequilibrium of Netdate that is occurring in cancer. So basically, if there's a pathway in cells that can detect that the Netdate has been upregulated, and how the system responds to this upregulation of Netdate. Now, so here, I mean, what we've done is we have the collaboration with a group of Malu Martinez Santar, which is not so far away from Santander. It's actually Sigby Ogun in Bilbao. And what Marina Serrano, a PhD student in Malu's lab, what she did is she actually monitored this upregulation of Netdate in cancers that I just described to you. So here, what the group of Malu have done is they created a hepatocellular carcinoma model system in animals, which is based on the deletion of this transcription factor over here. And these animals develop liver cancers, spontaneous liver cancers. And what Marina did is she analyzed, and this is a Western blot where you basically look the Netdate levels in either control animals or in tumors. And I think you can appreciate that in tumors, you have an increase of protein NEDylation, consistent with the idea that the Netdate pathway is upregulated in tumors. Now, what is interesting is that Marina we think that she found the molecular basis for this increase of Netdate in these tumors, because she found that specifically in the tumors that she created, the levels of the deconjugating enzyme NEDP1, they're completely gone. So basically, the enzyme that removes Netdate from substrates is deregulated in these tumors. And that now provides an explanation why in these tumors you have this accumulation of Netdate. Now, I can tell you that this, of course, happened. We see this in animals, in mouse model systems, but we can see this very similar thing, and we can recapitulate this in vitro, in tissue culture cell lines. And here I just show you one example of a cell line, of an osteosarcoma cell line, that if we if we make a CRISPR knockout of this denetylated enzyme, we see this massive accumulation of Netdate, very similar, if you like, to what we see in tumors. So clearly this enzyme is very important for the cycle of Netdate. And of course, if you delete it, you cause this accumulation of Netdate. And I can tell you that the proteomic studies, it told us that this accumulation you see here is really mainly the accumulation of poly-Netdate chains. So now, the key challenge that we had to find here is that what is accumulation of Netdate chains they're doing in cancer? So what is their role? So, and how can we deal with this? So in our lab, in order to understand this, in our lab, we're using as a model organism C. elegans, like the worms. And what we've done here is we make a knockout animal where we deleted the NETP1 gene from these animals. And what we found is that this enzyme, and if you like, the deletion of this enzyme, the accumulation of Netdate that I just showed you, has a very important role in the DNA damage response and apoptosis induction. So what you see here is you see the germline of the animal, and here you see the wild-type animals when they're exposed to ionizing radiation, and you see the accumulation of the so-called apoptotic corpse. So this is the induction of apoptosis in the germline of the animal, which is the physiological response upon DNA damage. Now, in the animals where we deleted the NETP1 gene, so in the conditions where we have a lot of NETs, like in tumors, DNA damage-induced apoptosis was completely blocked. So basically, there was no apoptosis whatsoever. And we recapitulate the same thing in human cells. So here you can see that if we in this case, if we do a knockdown experiment of NETP1, we compromise the induction of apoptosis measured by caspase reactivity, over here. So here we think that we found a highly conserved mechanism, a pathway that controls the apoptosis in cells. So the conclusion of these studies is the following: is that this enzyme NETP1, its major job is to constantly remove a Netdate from substrates upon DNA damage, and that is required for the DNA damage response to induce apoptosis in cells. Now, if you delete or if you eliminate this enzyme, you have accumulation of Netdate chains which somehow block apoptosis, which again, as I said, this recapitulates exactly what we see in tumors. Now, when you measure DNA damage, of course, you want to focus on the chromatin, and we know that these molecules play a very important role in how the DNA damage is signaled, if you like, in the cell. So, for example, ubiquitin chains or SUMO chains, they accumulate on the DNA damage sites where they sort of send a signal for either DNA repair, which can induce the cell cycle arrest in order for the system to have the time to repair the DNA damage. But if the damage is too much, it cannot be repaired, that will induce apoptosis in the cytoplasm. Okay, so this is a sort of our very basic knowledge of how DNA damage can either be repaired or eventually be sort of induced in apoptosis if the damage is too important. So we know that now that ubiquitin and SUMO, they directly go in the sites of the DNA damage response. Now, I can tell you, and I'm not going to show you all the data, that in our case, when we look in the accumulation of NET in NETP1 knockout cells, we didn't see any defect whatsoever at the chromatin level. So basically, the system, the DNA repair is absolutely fine, cell cycle arrest is absolutely fine, but still, somehow, these cells, they cannot induce apoptosis. So that told us that here we have a defect that is downstream of the whole signaling pathway over here, and it's specific at the sort of induction of apoptosis in the cytoplasm, which you will see that it end up to be, as we call it, a protein quality control defect. Now, here just highlight very briefly how apoptosis is induced in cells upon DNA damage in the cytoplasm. And a key factor here is this protein called APAF1. So this is a cytoplasmic protein, which is apoptosis inducing factor 1. And normally it exists in a monomeric state, but upon DNA damage, when the cell has to induce apoptosis, this protein gets oligomerized, and this provides a platform for all the caspases to come and bind to it, and then they get activated, and then they go and chop up the DNA in order to elicit the apoptosis in cells. Now, a key factor for this oligomerization business, if you like, that happens is the chaperone HP70. So HP70 is a chaperone that normally binds to APAF1, but in order for APAF1 to get oligomerized, HP70 has to be released. It has to leave. It has to be activated and released by monomeric APAF1 in order to allow the oligomerization of APAF1 and induction of apoptosis. It turns out that in NETP1 knockout cells, the defect, as I told you, is not at the chromatin level, but is indeed at this step. It basically, NETP1 controls how HP70 is released from FF1. So here, what you see is an experiment where we monitor the interaction of FF1 with HP70. So here you see the interaction of these two proteins. When you put DNA damage, in this case it's toposide, you get the release, as I said, the release of HP70 from FF1. But now if you look in cells where we have deleted NETP1, so basically we have a lot of Netdate, this release is basically blocked. So basically this concept, these two proteins, they are blocked, and we think that that's the way that somehow Netdate blocks, if you like, the formation of the apoptosome and eventually the induction of apoptosis. Now, how does this happen? We found that Netdate is directly binding to this protein HP70, but the outcome, if you like, of this interaction depends on the status of Netdate. So HP70 can bind both to Netdate chains, which, as I showed you, accumulate in tumors and accumulate when we knock out NETP1, but also binds to mononedate, to free Netdate. But depending on this state, the outcome of HP7 activity is totally different. So in the case that the Netdate is in a mono state, it can activate HP70, and that allows, as I said, the release of HP7 from APAF1 to induce the apoptosome. But when we have the accumulation of Netdate chains, this HP70 now is inactive. It is blocked in the sort of interaction, is blocked with the monomeric APAF1, and it does not allow the formation of the apoptosome and subsequently in the apoptosis. So now we think we understand of how Netdate chains can actually block the induction of apoptosis by blocking the activity of HP70 and its release from this protein called APAF1. So this is just a schematic to show you how we think that NETP1 is involved in the whole DNA damage process. Its key job of this enzyme is to remove, if you like, deconjugate Netdate chains into the free Netdate chains, with this sort of acting as a signal to activate the chaperone, which then allows the formation of apoptosome and induction of apoptosis. And we think that when you block this enzyme, you accumulate these Netdate chains, which basically block all this process, and then there's no apoptosis in cells. Now the question is this relevant to these tumors situation that I just showed you to do? So what we've done here is we went back into these tumor cells, okay, that if you remember, these cells that have reduced levels of NETP1 and they have a lot of Netdate. So what we've done is we put back the wild type NETP1 into these tumors and ask the question, can we now see apoptosis in these tumor cells? And this is now the experiment here. So here you see the induction of apoptosis in the normal tumor cells, in the control cells. And it says that we put a mutant form of NETP1 that is not active. And this is a sort of induction of apoptosis. But if we put back the wild type, so if you put now the wild type NETP1 in these tumors, you can restore apoptosis. So we think that this deregulation of NETP1 in these tumors might actually have as a tumor suppressor function. And we think that the formation of these tumors is because the decrease of NETP1 and the accumulation of Netdate. So in terms of therapeutics, NETP1, as you can realize, is not a good target, because inhibiting NETP1 will actually block the induction of apoptosis and will promote tumor growth. So it's not a good target. But if you remember, I told you at the beginning that this NETP1 is part of a cycle which depends on the activity of the E3 enzymes that they put Netdate on, and of course NETP1 that removes Netdate. So what we're trying to do at the moment is to find the E3 enzymes that they make these Netdate chains, that actually works with NETP1 in order to make these Netdate chains, because we think that these enzymes will actually be good therapeutic targets to inhibit the formation of Netdate in tumors, which in theory should increase and reduce apoptosis in these cells. So that was the first part, was like just to show you a bit of sort of basic biology of how sort of different components of the Netdate system can be involved in tumor progression and potential therapeutic targets. But now what I will do in the last 5 or 10 minutes, I will just show you a bit how potentially strategies that we can use the Netdate inhibitors in the clinics. Now, as I told you, the pathway is upregulated, and because of the success of the bortezomib, the people develop inhibitors for the Netdate system, which they block the E1 enzyme for Netdate, this compound called MLF124 or pevonedistat. So the compound is an AMP analog, so you can see the structure over here. It has a sulfonate group, which is different from AMP. And as I said, is in phase 3 clinical trials for multiple myeloma for solid tumors. Now, a key experiment that shows the specificity of this compound, because as you can realize, in all these clinical trials and all this development of chemotherapeutics, a big question is how specific are these compounds that we're using in order to target and inhibit the protein of interest that we want to target in cancers. So what people have done with this compound is to treat cell lines with very low doses of the compound in order to develop cell lines that they become resistant to MLF494. And then what they've done is they took these cells that they're resistant to MLF494 and they sequence the genome in order to find what type of mutations these cells acquired that cause the resistance to this compound. And what is very interesting is that in this sort of analysis, what they found is that the tumor cells that are now resistant to MLF494, they make mutations in the E1 enzyme where the compound binds. So now this mutation in the E1 enzyme now makes the compound that it doesn't bind to the E1 enzyme anymore. So the enzyme is still active, but now the MLF194 cannot bind to the E1 enzyme because of this mutation. And that's how these tumors now, they cannot see MLF194. But what is critical, it really shows that all the anti-tumor effects that we see of this compound is because of the inhibition of this enzyme. Okay, so that's very critical, if you like, experiment that shows the specificity of this enzyme in order to induce these anti-tumor effects through inhibition of the Netdate pathway and the Netdate E1 enzyme. Now, basically, and sort of a thing that scientists always look when they test chemotherapeutics is that they induce the activation of tumor suppressors, because in many ways, that's what you want to do in tumor cells in order to activate tumor suppressors to actually kill the tumor cells. And a key tumor suppressor that scientists always look at for its activation is a p53 tumor suppressor that I guess you all know, and it's a tumor suppressor that is mutated in almost 50% of tumors. So of course, the rest of the tumors that have wild type p53, the compounds that they will induce the activation of p53 will be something good in order to eliminate or to kill these tumors. So MLF494 indeed activates p53. So that was looked at the moment promising, and that looked something good because that was, if you like, give us confidence that by activating tumor suppressors like p53, MLF494 can induce apoptosis and kill tumor cells. Now, I'm not going to go into details, but just to show you that now we know the mechanism of how MLF494 really activates p53, and I will just mention that it involves the nucleolus into the nucleus, this compartment for which p53 is very sensitive. So now the question that we want to address is now we saw that MLF494 activates p53. What we wanted to now to understand is that how important is this activation of p53 for the MLM, for this compound to kill cancer cells. So what you normally do in this type of situations to test this hypothesis is we have a group of isogenic cells, which they have either wild type p53, the colon cancer cell lines, and they have wild type p53 or no p53 at all. And then the idea is that you apply the compound in both cells, you measure the viability of the cells, and actually then you can tell whether the activation of p53 is very important for the killing of these tumor cells. And indeed, Lara, PhD student in the lab, that's what she did over here. So here you see the wild type p53 cells, when treated with this compound, the survival really declines in a time-dependent manner. So indeed, sort of that was promising that the compound has its anti-tumor effects. Now, the big surprise though came when we looked into p53 negative cells. So here we were expecting that the p53 negative cells will be resistant to this compound. But in contrast, what we saw is that these cells, they are actually more sensitive to the compound compared to the wild type p53 cells. So in contrast to what we believed, it looks that this activation of p53 that you see here by the compound is actually protecting the cells, not killing the cells. So that was quite intriguing. And what Lara wanted to do is wanted to try to understand why the activation of p53 was protecting these cells from cell death, at least to some extent. So what she did is she did a cell cycle analysis using again the p53 plus less and the minus cells. And here you can see that the application of the compound is quite toxic. So here's the normal cell cycle profile. And here the cells are really accumulating, arresting arrest phase, and they die from a sort of an S phase, block. And you can see here that the MLF194 is effective in both wild type cells and sort of p53 negative cells. But what Lara noticed is that specifically in the wild type p53 cells, this compound was also induced in a small G1 arrest that it was not seen in the mutant cells that was quantified over here. So Lara wanted to test the hypothesis if this G1 arrest somehow was protecting the cells from dying when applied upon application of the ML494. So in order to test this hypothesis, Lara applied the trick where she basically used a compound called actinomycin D. This is basically transcription inhibitor. But this compound, when you use it at a very low doses, this compound is using a cell cycle arrest, a G1 or G2 cell cycle arrest that is fully p53 dependent. Okay, so now you can use this compound and you specifically can block the cells at the G1 or the G2 and ask the hypothesis if now the MLM494, the Netdate inhibitor, will now be effective in these cells. So here, the experiment over here. So here, what she did is she pre-treated the cells, both the wild type and then minus the negative cells with actinomycin D, and you can see a G1 and G2 arrest in the wild type cells, but the negative cells, they keep cycling, okay? There's no p53, so actinomycin D cannot cause this arrest. And then after the pre-treatment, she applied now the Netdate inhibitor. And as you can see here in the cycling cells, the mutant cells, they are sensitive to the MLF194, but not the wild type p53 cells, because they are still arrested at the G1, G2 stage. And somehow this pre-treatment, it protected the cells against the Netdate inhibitor. Now, I will go this very fast, but actually we can see the same thing in survival assays. So here you can see that the compound is toxic both to wild type and negative, p53 wild type cells and negative cells. But if you do the pre-treatment with actinomycin D and then put MLF194, MLF194 kills only the mutant cells, but not the wild type cells. Okay. So we also wanted to test this in a vivo sort of situation. So for this, we use zebrafish as a model sort of organism. And what we did in this case, we treat these embryos with MLF194 alone or in combination with actinomycin D. And you can see here that when you treat with an Netdate inhibitor, the inhibitor is inducing toxicity. This is toxicity, caspase-3 activity, apoptosis in the sort of live embryo. But if you pre-treat with actinomycin D and then put the inhibitor, now these animals are totally protected. So in many ways, what we think we found is a strategy to protect healthy cells against the toxicity that is induced upon MLF194 by using this pre-treatment with actinomycin D. So now, why this is important for cancer therapy? Because in any sort of tumor situation, like for every patient, you have two types of cells. You have your healthy cells, which in this case they are wild type p53 positive, and you have mutant cells, tumor cells that, as I said, 50% of them they will be p53 negative. So by doing this trick, by pre-treating with in this case with actinomycin D with a compound that activates specifically p53 to cause a cell cycle arrest, you can block the cycle of wild type p53 cells of the healthy cells but not of the tumor cells. And now in the second phase, these tumor cells will be very sensitive to in this case, to the Netdate inhibitor, which will cause mitotic catastrophe and apoptosis, but not the wild type cells, which again, they will be sort of be able to recover once you remove the treatment. So in this case, we think that you can devise an approach to prevent any toxicity of this compound that will be inducing in the healthy cells in every patient. So, and this, of course, is because all the chemotherapy side effects, they are because of the activation of p53 in the healthy system. So this is what we want to try to prevent by doing this trick. So this concept, if you like, is called the p53 cyclotherapy approach, and this is to, if you like, to exploit the guardian of the genome to protect normal cells from the chemotherapy-induced toxicity. Now, the advantage that this gives you is now by protecting the healthy cells, you can really increase the dose of the compound in tumor cells. So if you like, the maximum tolerated dose, it can now be increased, because now by protecting the normal cells against the toxicity of the compound, you can increase the dose that you can use in order to specifically target the tumor cells and not the healthy cells. And I will stop here. It's just to thank the two key people that do this work. So Meric Bailey, he did all the work that I just showed you on NP1. Lara did all the experiments on the cyclotherapy and the use of the inhibitors to protect, if you like, wild-type healthy cells against the toxic effects of the inhibitor. And we had a fantastic collaboration with a group of Manuel Martinez Santana in Bilbao, where she did all the work on hepatocarcinoma and NP1. And the work in zebrafish that I just showed you, we did with a group here in CRBM, Benedict de Laval, on the effects of the cyclotherapy. So I stop here. Thanks very much for the invitation. I hope it was relatively clear, and I will be very happy to take any questions now. Thank you very much.
Thank you very much, Dimitris. Well, we have some time for questions now. I don't know if someone wants to. Well, I will start then. Dimitris, I have some basic questions here. I would like to, do you have any, or someone has any idea of what is the percentage of, what percentage of Netdate action is in a certain pathology could be attributable to its ubiquitination regulation, and what is due to the regulation of other proteins? Is there any idea in a certain pathology?
No, so it's. I mean, I think what is pretty clear is that the role of NEDs in controlling the, as I mentioned, the ubiquitin system and the calling ligases, it's the major role of NED. So, but I have to say that from experiments that we've done, it doesn't always explain everything. So we found sort of particular cases or types of tumor cells that they're sensitive to all the non-calling targets that we just look. For example, in hepatocellular carcinoma that I just showed you, this accumulation of Netdate has nothing to do with callins, and still it looks that is important for the tumor development. So clearly, all this calling aspect that I just showed you is the major part by which Netdate operates, but doesn't explain everything.
Okay. Someone has any question? I don't know. I'm just checking the chat here, but I don't know if there's someone else. Oh, there's someone. Monica, go ahead. You want Monica?
Hola.
Oh, okay.
Hola.
Good morning. Thank you very much. I just have to go to class, so this is a very quick question. Thank you for your excellent introduction. I really enjoyed it, because I usually teach this in the medical schools, and for me it's been really enlightening. I only have a basic question regarding chaperones. You talked about HSP70, that it seems that it's a chaperone that is involved absolutely in every process in the cell, really critical chaperone. And this is really new to me. But I was wondering, do you know if HSP90, that is often overexpressed in cancer, has any role in proteasome degradation like HSP70?
No, clearly it's been. I mean, I didn't talk about HSP90 here, but clearly HSP90, it's also involved in. And normally they're working together with HSP70. So these two chaperones, they are the major chaperones in the system that they working together in terms to degrade misfolded proteins. So what is very interesting is that you see a very strong cooperation between these two proteins. So for example, when you block the activity of HSP70, the system upregulates HSP90 and vice versa. So there is a very close interdependency on these two, on the function of these two chaperones, but while they both the both of them involved in the degradation, if you like, of misfolded proteins, they seem to have specific targets. So each one, like they have defined group of proteins that they target each individual ones. And now there's actually studies in different type of cancers where they show that the deregulation of either HSP70 or HSP90 can have a completely different outcome in cancer progression. So yes, these two proteins are working together. In some cases, yes, they might, controlling sort of similar substrates. But what is also clear is that they have distinct sort of functions to control protein degradation and group of proteins for degradation, and their sort of role in tumor progression.
And if I may ask another one, if instead of cancer we talk about neurodegenerating cells that have aggregates of amyloid, do you know if anybody has explored the role of either HSP70 or in particular HSP90 in the disassembly, degradation, or whatever, a way to treat all these aggregates of amyloid intracellularly?
Yes, so what is absolutely clear now is that especially HSP70 is absolutely essential for the formation of these toxic aggregates into the cell, but also for their elimination. So these aggregates that we see in neurons, they have different sort of phases and qualities. So there are aggregates that they are formed, but sometimes they can be sort of eliminated, they can be reversed, they can be eliminated by the system. But there are other type of aggregates that they are completely irreversible and they just stay there forever. Now, the ones that are getting the reversible ones, the elimination strictly depends on HSP70. So of course, when you block HSP70, you make these aggregates now to becoming irreversible. Mm-hmm. So, I mean, it's not still very clear, of course, if HSP70 is a good therapeutic target for these cases. Maybe probably not, because if you block HSP70, most likely you will induce aggregation of proteins. But what's absolutely clear is that HSP70 is essential for the formation of aggregates in these pathologies.
Well, thank you very much.
Thank you very much.
Thanks for your talk. I really enjoyed it. It's been brilliant. I really loved your models and your slides. Very enlightening. Thank you.
Thank you very much. Thank you.
Sorry, I have to go now to class. Bye-bye. Thanks.
Bye.
We are very jealous about your Western blots. We would like to have those, those nice Western blots to show. Well, do you have any questions? Someone else? Well, I have another one, just some basic one too. Do you know how shifting is the nedilome? I mean, it's something like when you extract proteins from certain cells or certain pathology, you extract the proteins and you have to add, anti-kinases, anti-phosphatases. Is this the case for nedylation? Is it that shifting since it's a reversible process?
Yes, so actually, so these pathways, we think that they are even more sensitive compared to phosphorylation in terms of removing and losing the modification. So the deconjugating enzymes, they are extremely active. So if you don't take any precautions to inhibit these enzymes, when you lyse the cells, everything will disappear. And actually, in many ways, that's one of the reasons why ubiquitin, as a post-translational modification, was really much later came in the field, like in terms of how many proteins they get in ubiquitin, because in the past, when people were doing Western blot analysis, they were lysing the cells without any inhibitors, without anything, and of course, all the modifications were disappearing. So we never used to see ubiquitin, modifications in the past because of this reason. And once you put inhibitors, I mean, the whole picture completely changes.
Okay. Mm-hmm. So, any more questions? Well, I don't know if there's some other question in the other chat.
No, there is none.
Nothing?
Nothing.
Okay. So, well, maybe we should stop here. Thank you, Dimitris, for your nice
Okay,
talk.
thanks again to all of you. Thanks for listening.
We'll keep in touch. Yeah.
Keep it that.
Okay. Thank you, everyone, and bye.
Bye-bye. Thanks.